So welcome everyone. I think we can start. It's about 3 minutes past the hour. Welcome everyone to the SCAI Kairntech webinar on knowledge graph creation maintenance from biomedical content. Glad that so many of you made it to the webinar. And this is what we have in mind. This is what we have prepared for you. There will be two presenters today. There's going to be Professor Martin Hofmann-Apitius, the head of bioinformatics from Fraunhofer SCAI in St. Augustin, and myself, Stefan Geisler from Kairntech. And this is what we have prepared for you. Martin will start talking about the construction of knowledge graphs from biomedical content, a topic that they have been working on at SCAI for quite some time now. Martin will then hand over to me, and we from Kairntech will describe how we are using AI and NLP methods to automate the creation of knowledge graphs from content analysis. And then Martin will conclude, placing these efforts into the larger context of ongoing projects at Fraunhofer SCAI, wrapping up what we have learned, maybe an outlook to next steps. And then we have reserved time for your questions, trying to get into a discussion, see what we have learned, what maybe next steps from what we have seen. And with that being said, I pass on the word to you, Martin. The floor is yours.
One of the key questions was always, yeah, you invest a lot of effort into generating highly curated pathophysiology mechanism graphs. They are wonderful tools, but how do you keep them up to date? This is it sounds like a trivial thing, but it is actually not. And we have recently tackled that point together with Kairntech. And this is essentially the story of what we do, or what we did. People who know me a little know that we go for bigger pictures. We follow indication-wide approaches in our work, and the philosophy behind that is essentially that based on the experience I made when we were doing still genome sequences, you know, those people who are a bit older remember how impressed we were by the sheer size of the human genome. And I remember a talk 3 months before the human genome draft, the first draft was published, by a colleague from Insight, who said there must be at least 140,000 human genes. So big numbers, and we know that in entire indication areas, the data falling around that are available are complex. They are diverse. They are really of heterogeneous quality, I tell you, and everybody here probably knows that. They are scattered. They are not easily to obtain very often, and if you get them, you may not be allowed to work with them. So there's a lot of obstacles, but still the number of data sets for an entire indication area is huge sometimes, but it's not infinite. And the same is actually true for knowledge. We are in the situation now where we see that there's complex knowledge, diverse, scattered, contradicting everything, poor papers, good quality work, and I'm not daring to judge on how much is wrong in PubMed and how much is right, but the tricky bit is to know what is the proportion that is right. So there is the same situation when it comes to knowledge. It is scattered, it's incomplete, contradictory, sometimes diverse, yet it can be formalized. We have now the technology at hand to actually bring knowledge into computable graph formats. And data and knowledge in any given indication area actually can be ordered, quality assessed, and curated. We have been spending the last 10, 15 years on that, doing that in the area of neurodegenerative diseases and psychiatry. This is two large indication areas that we focus on. And data and knowledge can be made interoperable and modeled across scales. And we can subsequently compute on the combination of data and knowledge. And that is what we in my department try to do for some while. And I think we have been quite successful with that. as I show you in the next slides. So work in our department is organized along a value chain, as we call it, or a problem-solving workflow. It starts typically with unstructured information mining. Then we have a strong activity in knowledge graph technologies. We combine models, or we use models to combine data and knowledge and have algorithms that act on the interoperable representation of data and knowledge, and a strong AI group in our department is actually turning that into actionable insights and decision support. We have been working on shared semantics, and probably one or the other amongst you knows that already. Most recently, we have been collaborating with the FDA, the NCBI, and the University of Michigan to update the clinical trial ontology, which they at the NCBI want to use to link PubChem with clinicaltrials.gov. So integrative data semantics, so to speak. And we have been working on an entire framework, semantic framework for neurology in a wider sense. Over the last years, you see first publications, 2013 and so on, almost 10 years ago. And very recently, we addressed even the pandemic with a COVID-19 multilingual ontology. So that is our background in semantics, and formal semantics knowledge representations. We are making widely use of information extraction technologies to create knowledge graphs, which are specific for a knowledge domain. And the syntax or the technology we are still using for that is called BEL. Ted Slater is in the call here. He's one of the early birds in adoption of BEL, biological expression language. And that allows us, this language, this formalism allows us to encode a complex sentence like this one here, this phosphorylation of glycogen, synthase kinase 3 beta at threonine 668, increases the degradation of amyloid precursor protein. So a typical sentence that you find in literature about, in older literature about Alzheimer, and the coding of that complex expression here, the formal representation in BEL looks like this here. So the phosphorylation of GSK3 beta at the modification site, at the threonine site, leads to the degradation. And you see that you can actually read BEL as a human, and you can read BEL as a machine. And that's the nice thing. BEL is a formalism that is comparably easy to learn, and it's comparably low complexity, which allows you to use it it's quite handy. It's not, expressiveness is of course limited because of the limited number of relationships that BEL knows. What have we done so far in the field of neurodegeneration? We have actually created the largest computable cause and effect model for Alzheimer worldwide. This was work by Alpha Tom Kodamolil in 2015. Later on, Daniel Domingo Fernandez and our team has generated a series of subgraphs for Alzheimer, 124 subgraphs encoding pathophysiology mechanisms, and he even designed a mechanism enrichment server for that. The beauty about BEL actually is that it spans, it is agnostic when it comes to the type of information you encode. So BEL can capture and represent knowledge about that is dealing at the genetic level, genetic variation, proteins, enzymes, small molecules interacting, entire biological processes, and up to the clinical level. We have graphs where we have imaging readouts and cognition tests represented in the same graph and linking to a variation in a certain genetic key, in the genetics of a key protein or gene. So that's BEL, that's our world that we use for knowledge graphs. That's the syntax, and there's an entire Bell framework behind our work. Now, one of the things that we have been focusing on in the COVID context, actually, and predominantly in the Alzheimer context, is to use these sort of graphs for the identification and prediction of drug repurposing candidates. That's actually a story that we published this year in a specific context of Alzheimer, and one of the hallmarks, the p-tau protein, which is one of the key hallmarks of neurodegenerative diseases, both in Parkinsonism and in Alzheimer. We have used all our knowledge graphs. We have an entire framework, the eBell framework, which is a Python suite to handle Bell and to do link outs and integration of external knowledge from curated databases that can be genetic variation databases, pathway databases, chemistry databases. You see a couple of databases here like DrugBank and Stitch and TTD. And by enrichment of the knowledge graphs, the pathophysiology mechanism knowledge graph, with chemistry and drug target information, we establish what we call a pharmacome. And the human brain pharmacome is a gigantic drug target mechanism network that we have generated with a focus on neurodegenerative diseases. So it contains a lot of knowledge encoded as a graph, as a cause and effect graph on Parkinson, Alzheimer, epilepsy, FTD, ataxia, and even multiple sclerosis. And the entire thing is heavily enriched with drug target information, so ligands. This is just statistics on the numbers. You see that we have 11,000 genes in there and so on, but this is just big numbers and it's a big hairball. Yeah, we know those things. The good thing is we can handle them quite well in the graph database that is underlying our analysis framework. Now, more important is this one here. This is the p-tau mechanism subgraph, and the people who are a bit familiar with neurodegeneration know that p-tau is one of the key players. You find the protein in a misfolded state in fibrillar tangles that are present in the brains of people suffering from both Parkinson and Alzheimer and mixed pathologies. This interaction network here shows about the size of the complexity just around the biology of tau. And we were in particularly interested in drugs that regulate the post-translational modification of tau. So we have tau as one of the key players, and a lot of people are interested in this key player. We know that it's heavily modified by post-translational modification, including phosphorylation events. And we were interested in drugs that actually modify or modulate the post-translational modification, in particular phosphorylation of tau. And that is the p-tau specific pharmacome graph with the chemistry and the drug target relationships included. So what we did, and that is a very brief version of what you can read in the paper by Vanessa Lager-Ruprecht and others that we published this year. We actually looked in the graph database for upstream controllers of the tau phosphorylation. We collected all the nodes, but also identified all the druggable nodes that are potential controllers of tau phosphorylation. We did additional text analytics to expand on some of that space and update the space for one reason that I show you here. We looked for the upstream modulators and controllers of tau phosphorylation, but some of them were not druggable, and some of them actually had a relationship which had intermediates, and we identified those intermediates by additional literature mining. a lot of that work was done using text mining technologies, but also a lot of manual expert curation. We have a very strong partnership with a curation company in India, started by one of our co-workers, Alpha Tom Kodamolin, and they help us a lot when it comes to high quality graphs. And to cut that story short and to tell you how we work then with the potential drug target information in that graph. Here's a screen that we derived directly from the pharmacome tau pharmacome mining. We identified HDAC6 as a potential target of the modulation of phosphorylation of p-tau. We did an HDAC6 discriminatory screening, dissociating the histone-specific inhibition from the Alzheimer context. So the cancer drug type of HDAC6 activities and the neurodegeneration type of activities, and came up with a variety of hits that have been confirmed also with respect to their modulation capacity for p-tau modulation. So I'm not going into too much detail on the pharmacology here. I just want to show you that a highly curated cause and effect graph around one of the hallmarks of neurodegeneration gave us the power to make a decision on screenings with a repurposing library of 6,500 compounds that we routinely use. We means in that case our colleagues at Fraunhofer ITMP in Hamburg. They are screening specialists. So, and that was the situation that we faced, and now we thought we need to do a update of the tau graph, because the knowledge that I've shown you, the graph I've shown you, is now in the meanwhile 1.5 years old. And in the meanwhile, we have seen a lot of relevant literature coming up in new and interesting edges and nodes appearing in our graph. And the question was, how do we keep that up to date? I told you before that we have, just for Alzheimer, 124 of those graphs. And if you want to keep them really up to date, this is a hell of work, and that needs to be automated. And that is actually where I hand over to Stefan, because this is where Kairntech came into the game. And I should maybe say that we teamed up with Kairntech because we have been working on relationship extraction for a while, and we have seen that they are specializing into such a degree that it actually makes a lot of sense if we join forces, and that's actually what we did. So now I need to see my cursor, sorry, and stop the screening, the sharing. So, Stefan, now.
Thanks a lot. And now we are back. So where are we? About here. All right. So thanks a lot, Martin. We heard a lot about biology and pharmacology now. So how does NLP come in here? That's our part. So natural language processing is what we are focusing on at Kairntech. Kairntech is now in the 4th year of its existence. It's a French startup specializing on the combination of NLP and AI from its headquarters in France and in Paris and close to Heidelberg in Germany. We apply these methods not only in the life sciences, but also in, for instance, the analysis of press documents with the French government on other topics, and with the analysis of financial information. So for us, the analysis of documents in the life sciences is one important, but one use case where we apply our methods. And our mission is to apply these methods such that they can be applied by domain experts. So people who know what the topics in their domain is, but who are not necessarily themselves programmers or data scientists. A phrase that has come to be known in this regard is no code or low code. So that means you can manipulate sophisticated machine learning algorithms without actually programming them. And that's our mission. So for the project where we came into play, Martin and I have known each other for, I don't know, 15 years or more. And suddenly, more or less out of the blue, in spring 2021, last year, SCAI came along and said, hey, we are on this project here that Martin just explained. And we need something that is able to extract from a large amount of literature information about entities, and most importantly, also about the relations between these entities on a specific corpus on psychiatric disorder. So can you do that at Kairntech? And we said, yes, sure, we can do that. That's our expertise. That's what we are here for. So we sat down and implemented on SCAI's requirements a pilot until about September 2021, and handed over the results to SCAI for them to assess what we have done. And apparently they liked what we have sent over. So since then we have sat down and said, this is looking good. So let's formalize the way how we can think about driving that further. We have laid down a few principles and plans in the memorandum of understanding. We have started some joint publication efforts. We have deployed our software, I'll come to that later, at the SCAI high performance computing cluster. So at Kairntech, we have a few isolated machines, but nothing that even comes close to the computing power that SCAI has available. And if you want to scale these things up, this comes in handy. So this is ongoing work right now. And we are preparing and working on joint activities like this webinar where we are in right now. We do all that on the basis of a piece of software that we used in this pilot and in all the other engagements that I mentioned earlier, that allows to, if you follow this sequence at the top, to import documents, either annotated or non-annotated, and then create a training data set from the data, training, optimizing, evaluating machine learning algorithms on this data, and then combine all that into a pipeline that solves a certain requirement from a client, from a project. And this can be done either graphically via a web front end, where we have a little screenshot here, or via the REST API that allows to integrate all this machinery into third-party environments and components. And this pipeline now is the place where we can implement, without reinventing the wheel for each new project, even sophisticated requirements as the one we got from SCAI, which said that we first need to recognize a number of entities, obviously, so proteins, genes, chemical substances, diseases, and things like that. For that, we were able to take out of the off the shelf something we have ready-made and we employ in a lot of projects, an entity recognition component that performs large-scale entity recognition on just about any topic, multilingual, that is constantly updated as the world around us is changing, and that links the recognized entities back to background information. So it's not only a string that is recognized, but really an entity that knows its position in a taxonomy and so on and so forth. We had to do some refinement, some housekeeping, some filtering. That is also something that has a defined place in the pipeline. We then computed additional entity properties. So not only, for instance, if we look at proteins, a protein was recognized, but it was recognized, is that protein in its original or in a modified form, like, for instance, is it phosphorylated? We did that by training a model that performs this computation and put it into this pipeline here. We then built relations between these entities, and ultimately SCAI needed all this information not in just any XML or JSON format, but in the Bell format that Martin explained to us. So also for this kind of output formatting, the pipeline has a place. So we were able to use our platform without too much changes, just by customizing the various steps towards the question that SCAI had asked us. Obviously, the entities are highly specific. So for proteins, they needed to be placed into the namespace HGNC.
for pathologies, diseases. MESH was a good fit for the drug and chemical abundances. We use JB for the biological processes, and they are referred to the namespace for gene ontology. And again, the entities that I'm talking about here are not only recognized, but they are disambiguated. In earlier years, in an earlier life, that would have meant that a lot of specific rule writing and definition of constraints would have to be written. Here, all that is happening via machine learning that is able to say, okay, I have recognized cancer, but am I talking about the animal or the disease? That's a pretty trivial kind of ambiguity. But as soon as you're working with non-trivial vocabularies, we all know that thousands of these ambiguities take place, and they are all disambiguated via machine learning. They are scored. Is this entity core, or is it just peripheral to this document? So they receive a numeric score. They are normalized, so each synonym is mapped to the preferred form in order to enhance searchability and findability. And finally, they are linked. What do I mean by linked? So here we have a screenshot of a piece of text where a lot of entities are recognized. And we see that this toll-like receptor number 3 is not only recognized as a protein, but by virtue of having its address, an identifier that links back to its database, knows what kind of entity we are talking about. You can, if you want, if your application benefits from that, you can present a little picture and other properties of that guy, because it has been linked to background knowledge, which requires disambiguation first, of course. After all this has taken place, we needed to construct relations between the entities that we had recognized. We did that on the basis of a training corpus provided by Fraunhofer SCAI. As with all non-trivial training corpus of that type, it contains a lot of relation types that occur often enough in order to be reasonably trained upon. So like increase, decrease, regulate, and a few others. They occurred often enough in order to run a training algorithm. For the ones that are not, we had to apply a threshold. And then most importantly, we had to also define an artificial relation, no relation, for all the 2 tuples of entities that are not in a relation. And we trained this component inside our pipeline on this corpus and handed over the results to SCAI, saying, so this is our results, please let us know what you think about that. SCAI took these results and sat down and evaluated what we had delivered, not the totality, but a decently sized sample of the results, and came back with the information that about 3/4 of the results make sense, which in relation extraction is a good value. So this was, let's say, one of the factors that led to the continuation of this kind of exercise. You see here a little graphical interface that allows a user to either place a little piece of text on the left or a file listing file identifiers, like for instance PubMed identifiers, into the application and call for the analysis. And what comes out at the bottom is BEL. So that's actually a list of BEL statements. That's always a subject that we have here, and the position of the subject in the text, and then a relationship, and then the same for the objects, and a couple of metadata that we are not filling here, but maybe the sentence is important. So these are BEL statements, and that is actually what we had, what our task in the project was, what needed to be constructed, and what we had returned then to SCAI. We were talking about knowledge graphs. So obviously there are nice graph visualizations here. Here we have one that we didn't build ourselves. Obviously, we have imported the data into a Neo4j database. So we have translated the BEL statements into the Cypher language for Neo4j. And then we asked this huge graph, can you show me, for instance, on this sub set of entities of results, show me a couple of proteins that are known by virtue of this graph, to be positively correlated both with schizophrenia and bipolar disorders. And we get a decently small graph of proteins that seem to exhibit that property. And if we focus on one of them, for instance, and ask it, please tell me more about that link here, the link knows where it is coming from which PubMed ID, and what was the sentence that led to it. And if you are curious and say, is that really true, then you can look up in the literature. There's ample evidence that this protein has this property of being associated to both these disorders. So that was the pilot that we had done on Martin's, on SCAI's request. The ongoing work since then is that in the pilot, it was not important how fast this analysis ran. So effectively, it was no problem to run this for a couple of days. But if you want to scale that up, if you want to do that regularly, if you want to do it on a much, much larger basis, it makes sense to think about speeding that up. And here it comes handy into play that SCAI is not only an expert in the bioinformatics around these questions, but also sits on top of a large computing cluster, and also has the expertise, has the colleagues in the team that know what it means to take an algorithm that runs on a single isolated machine and parallelize that so that it makes use of not only 1, 2, 3, 4, 16, but maybe of a couple of thousand CPUs. So that is a profession in its own, and the SCAI team has the expertise to do that. So they are right now working on transforming the machinery that I have described into a cluster-ready kind of analysis system. Work is also going on to extend the pilot question about finding relations on the isolated topic of psychiatric diseases, schizophrenia and bipolar disorders, to other therapeutic areas. Since we concluded that this makes sense, that this works, we are defining a joint approach and offering for third-party users, for third-party industry use cases on, for instance, computing topic-specific knowledge graphs for maybe other kind of areas, or updating them. So Martin explained that once you have the graph, the moment you sit back and people continue to publish, your graph will quickly age, will quickly become outdated. And the question is how to update that. And automatic analysis of large amounts of literature is obviously one way to proceed here. And then why not aim high? So this may be a bit of science fiction, but we have added that here for the sake of completeness. If all that works out, if the speeding up of the analysis to 2, 3, 4, 5 orders of magnitude more in computing power works out, maybe one day we are in a position to say, let's apply that on a decently large chunk, and maybe ultimately sometime down the road on all of Medline. Why not? Again, let's aim high. The moment you have automatic procedures performing that, suddenly these things become imaginable. So what we've seen in this exercise, we have seen that even on this very highly specific requirement, we were able to pull off the shelf components that are part of our software stack, for instance, for entity recognition, that perform reasonably well in this highly specific subdomain. We found that the notion of our processing pipelines is a fine place to implement all the little steps that were required in order to solve the tasks that Martin has asked us to solve. So the entity recognition, the housekeeping, the analysis of the specific properties of the entities, and then ultimately the construction of the relations and the encoding into the syntax that was required for the project. We were happy to get good feedback from the team at SCAI after we had submitted our results. And we jointly conclude that it is feasible to turn the knowledge hidden in large amounts of literature by automatic means into a machine-readable, machine-analyzable, machine-processable knowledge graph and that is a good, let's say, finding a good conclusion for that part of the project. And with that being said, I hand over to Martin back again to conclude his part of the presentation. So I'm stopping sharing. Martin, the floor is yours again.
Okay, thanks, Stefan. So you see my slide?
Yes.
Okay, so what did we see so far? I motivated this point of updating graphs, and I will show you where we stand there in a minute. And Stefan was also alluding to the proof of concept. So we did this initial, test with Kairntech, where we want to see. We know what our text mining machineries at SCAI can do, and we knew that Kairntech has this more modern approach, and we just wanted to have a good idea on the performance, and it's absolutely a non-trivial thing to extract from text and directly encode in Bell. I mean, those people like Ted, who is in the call, who know that world a little, know also that there's very famous text mining labs that did not manage to get that done in the past. We built our machinery, and we know, of course, of colleagues at Harvard, the Indra team, Peter Sorger's lab, who are using some ensembles of rule-based machines and so on to come up with informative triples from text. As Stefan already outlined, we will be going for indication-wide knowledge graphs and even the entire PubMed graph, which is still something that I would like to see become manifest before I retire. But now before I start talking about retirement, let's just look at the really concrete thing, that the real stuff that we have been doing now with the pipeline running on our cluster so far. We have been starting just a few weeks ago to operate that system, and we can do it in our hand. It runs well. And we are back to the unstructured information mining, this time addressing this topic or this challenge of how do we keep a knowledge graph up to date. So typically we produce knowledge snapshots. In the past, we have done cause and effect graphs representing some well-established knowledge, typically in a given indication area. But if we want to have this really continuously updated, how would that work? And this is now work that is largely also done by Negin Babaya and Sumit Madan and Mark Jacobs in our team. And Hassan El Said, I shouldn't forget Hassan. And from the names, you see already how international our team is. We have invested in corpus generation when you try to update a knowledge graph, you may want to widen the scope. You may want to actually be open for new, interesting, cool things. And that means with just a keyword search that you may have used before or so, this is maybe not the best idea. So we have invested in the corpus generation. Then we had to get the Kairntech workflow up and running, which was with the help of Kairntech done in a short while. And of course, we do some performance analysis. What you see here in this slide actually is how the literature on tau grows over time, and you see this molecule gets more and more attention in our community. So there is a steep increase in relevant knowledge. Now, when we define corpora, we have to balance between novelty and relevance, and we need to somehow have a clue on where's the transitive hull, where's the scope, the delineations of the scope for this particular topic. And And therefore, we tested and optimized a variety of CAN queries and looked into details of the literature, whether it really fulfills the expert criteria of relevance. This is something that you still need to do with people who are experienced in the field. We have done some characterization of corpora and finding a rational, reproducible rational for what we call relevance. And we used a variety of approaches like n-grams, TF-IDs, and mesh and profiles essentially to play around with retrieval of relevant corpora. Then, after defining actually a variety of corpora, we didn't do that just with one. We didn't believe that we have everything that is relevant just in one type of query. So we actually used a variety and a multitude of corpora that we generated. They are all dealing with the post-translational modification, phosphorylation, and modulation and control topic. We didn't do any topic maps so far, just to say that. That's on the agenda. Then we run the Kairntech workflow, and as Stefan already mentioned, we have something in average between 70 and 80% F-score. I'm still from the time when we were happy at the entity recognition level when we reached 80%. So I think for relationship, for triple recognition, this is outstanding. This is, maybe I'm getting old, but you know, for me, it's really something that I appreciate really. And then the question was, what we get in return is, of course, a long lists of triples, and that needs to be compared and identified as novel, as interesting, as relevant, and within scope. And that's something we do in our BigMe environment. This BigMe stands for a biological knowledge miner, which is a graph database backend plus a visualization front end, plus the ability to import all sorts of graph formats and do graph queries and so on. So here's the most recent visualization and update on what we have done with the tau graph so far. This is how it looks in a simple visualization in the state as we had it. You see a couple of high centers here, or nodes that are central, very highly connected, and these are typically tau itself and key tau features. We have here what the Kairntech pipeline delivered in addition so far, and that is actually a overlay of the two graphs. You see that we are in the range of about 15% it is of new triples that we have found so far. We looked into some of the biology, and thanks to Negin, she was doing a great job in that field. Some of the newly incoming triples are interesting because they represent a new wave, a new hot candidate of controlling neuroinflammation, so-called TREM2 protein. For those who are a bit in the biology of neurodegeneration, TREM2 not really surprising for those people in the field, but it shows that with the relevance criteria, actually we have a good, we actually had defined the right corpora, and you see that within, let's say, 2 years of time, we have a triple increase representing some knowledge increase of about 20%. Here's the references if you want to look into more details. The entire human brain pharmacome and tau drug repurposing story appeared in Cell Patterns recently. So that's a more recent story. Then we have done a similar approach in COVID-19, which appeared in scientific reports last year. We have been using knowledge graphs in attention models for the enrichment of graphs in literature, or the literature-based enrichment of existing graphs in a purely AI transformer-based approach, which is non-practicable, I should say, where if it only works with so huge amounts of graphs and text that it's not helpful when you go really into a specific biology, as we have seen it with the tau graph. So as nice as Helena Ballevin's work is, this is something that we would not, that route we would not follow for the updating of graphs, particular disease knowledge graphs. And yeah, COVID-19 work I don't cover here. So what I want to give you as a take-home message, and Josef, I've seen your hand, you raised your hand. I'll come back to you, and we'll should discuss all that. Knowledge graphs have proven really useful for the representation of complex knowledge about disease pathology in computable formats. This is something that we know now works really well. We have been using that for a lot of things like comorbidity mechanisms, pharmaceutical drug mechanism reconstruction, stuff like that. Here we have used it for drug repurposing. There's a almost endless space of applicability of these knowledge graphs. What Stefan has actually impressively demonstrated with his team at Kairntech is that AI on text actually is able to learn embellished statements. We will need to work on making that better. I mean, retraining and so on. So that we are not far from having reached the optimum. But for a first go, you know, in a almost indication-wide approach, the proof of concept experiment that Stefan mentioned on bipolar disorder and schizophrenia, that was way better than our expectation, well, my expectations were at least. And as I said, I mean, they produce Bell-conformed triples and output, which, by the way, have full provenance. So you can always go back to the source, always back to the sentence, and you have full evidence backtracing for each statement. We have demonstrated that you can use this approach to update pre-existing highly curated pathophysiology graphs used for disease understanding and the prediction of drug repurposing candidates. And now our next big thing will be, of course, we do the in detailed analysis of the updating of graphs, and that will be Negin's first paper. But the second is already underway, where we do indication-wide knowledge graphs. Besides the bipolar disorder and schizophrenia graph, we will produce a large depression network. And that will also be the subject of our next webinar, probably in the beginning of next year or so. And with that, I'm done.
That sounded like a good concluding remark. Things move. If you look back for a couple of years, a lot of the things that we were talking about would not have been possible. There's definitely scientific progress and technical progress in processing these kind of things, and we are happy to see that we are part of it, and this raises interest. As Martin has announced, we will continue to work on these topics. This is an exciting area, and your interest underlines that this rings a bell out at third parties from wherever you come from. So you will hear from us. You will certainly get the slides after this presentation. And from time to time, you will get invitations on subsequent events of this type. You will see publication efforts. So Martin has shown how busy they are on these topics. And unless there are additional questions, I think we can call it a day. Thank you all for attending, for your interest, for the discussion, for sticking out and hanging in for so long. And we keep in touch. Martin, a special thanks goes to you and your team. It's, as in the past, always a pleasure to work with you. You provide exciting questions and challenging tasks, and we are looking forward to driving that further. I think there's a lot of music, there's a lot of challenges still lying ahead, and we are happy to explore them with you.
Okay, sounds good.
All right.
Everybody have a good day, good evening, whatever you do.
All right. Bye-bye. Have a good afternoon.